There are billions of dollars and many companies working on it, and people will figure it out. We've found very small CRISPRs that allow us to move into the other organs like the CNS. So if we can go in and actually provide a one and done treatment to effectively allow them to just live a normal life, forget about their disease, that's the goal.
Our coverage of Meeting on the Med in Rome is kindly sponsored by Cellares. I'm Federico Cittrich, reporting for Onyx Live here in Rome at the Meeting on the Med. Today we took some time to have a chat with Devyn Smith, CEO of Arbor Biotechnologies. Devyn, good morning. Thank you for joining us.
Oh, pleasure to be here. I'm CEO of Arbor Biotechnologies. I've been CEO for 5 years. Prior to that was at another company in the Boston area and spent about a decade at Pfizer. Did some work in strategy consulting and a PhD back at the turn of the century. So, been in the space a long time. Arbor is a gene editing company. One of the unique features is the company was co-founded on an idea as opposed to IP. So the idea was, let's go out, find a lot of novel gene editing approaches that allow us to really tackle the entire scope of the genome, the entire scope of genetic diseases, so we can really create novel medicines for every type of disease.
What's the thinking behind that?
The initial focus has really been on pediatric diseases for the liver that are rare but devastating. So for example, our lead indication is in a disease called primary hyperoxaluria. It's a devastating disease. Children are diagnosed as toddlers with kidney stones and blood in their urine. And by the time they're teenagers or young adults, they have to have a kidney transplant. So there are treatments today, but it's basically treatment for life. So if we can go in and actually provide a one and done treatment to effectively allow them to just live a normal life, forget about their disease, that's the goal. So we're doing that in the liver. Outside the liver, it's really focused in the CNS space. ALS is a devastating disease. Patients are often diagnosed, relatively young, and it's a death sentence in a very few short years. So again, this is one where we're targeting, will be the first gene editing approach in the CNS, targeting ALS, so it's really breaking a lot of ground, and we hope will provide some great benefits for patients.
I think that most of the field has developed also from first generation CRISPR, so I wanted your take on how was that evolved until now, and also how that influenced the Arbor.
Yeah, so it's one of the benefits, I think, of science, is you get to build on the shoulders of those before you. So the first generation CRISPR used CRISPR Cas9. One of our co-founders is Feng Zhang, who is one of the founders of, and discoverers of the original CRISPR. And what we've done is, so Cas9 itself is a very large molecule, so you can really only limit yourself to the liver. We've found very small CRISPRs that allow us to move into the other organs like the CNS. So CRISPR Cas9, the way it binds to DNA is it has this sequence. It's kind of like a zip code. As you're old school, you write a letter and you mail it and it goes to a zip code. The zip code tells you where to go. CRISPR has a zip code, CRISPR Cas9. What we've done is expanded that and have a variety of zip codes so we can effectively hit anywhere in the DNA. So that allows us to have the breadth to touch anywhere in the DNA with a variety of tools. So CRISPR Cas9 traditionally has been used just to knock down, to remove something, a protein. We can do that. We can also remove. There's diseases that have these large repeats. You need to remove them, like Huntington's disease. There's other diseases where you want to rewrite a mutation and fix it, and so we can do that as well. So having that variety of technology sort of moves beyond that first generation CRISPR-Cas9 into the much more sophisticated editing.
MOMED is coming to an end. One of the key themes that were discussed during these 3 days was delivery. So I was going to ask, what's your opinion on that? How is that still influencing the field?
I think delivery, I think there's a couple of pieces to it. One, LNP, lipid nanoparticles used for delivery into the liver. That is pretty standard now, which is why gene editing companies are all focused in the liver on a relatively small number of diseases. The other place you can deliver is using AAV, so adeno-associated virus. We're using that in the CNS because you can target the central nervous system with AAV and deliver. Because our material is small, we can fit it into AAV and we can deliver to the AAV. But delivery is a key opportunity and challenge. I think there's, you can utilize AAV for some tissues where you can have specificity. There's other tissues where it remains to be seen what's going to be the best delivery modality. But luckily there are billions of dollars and many companies working on it, and people will figure it out.
Gene editing has overcome incredible problems and is going really fast. It's updating and evolving really fast, but there's still many things to, overcome, many problems to overcome. Do you think there has been too optimism at the beginning of the field about it?
Yeah, it's a good question. I think it's your typical, people probably heard the Gartner hype cycle, right? A lot of hype early on, then the reality sets in that things are harder than you think, and then it moves on from there. I think gene editing went through that. I think it is now in the stage of we're seeing real data. So earlier this week, Intellia read out their phase 3 study, which allows them to now begin to file for a drug approval, which would be the first in vivo drug approval in gene editing. This year alone, I think we'll have clinical data. I think 6 or 7 other companies will have clinical data. So now you're actually beginning to see the tangible results of all of this work on the benefits to patients across a variety of diseases. So we're very excited to see that. I think it's just the tip of the iceberg, as you said. I mean, maybe 1,000 to 2,000 patients have actually been dosed with an in vivo gene edit to date. And as you look ahead, as we move into bigger diseases, different patient populations, at some point, I think that vision of, someone will come in at a young age, get their DNA sequence, identify the right mutations, apply the right CRISPR, fix it, and then they don't have to worry about the disease. Today, we're treating symptomatically. In the future, we want to treat prophylactically. And so I think in some future generation, it'll be like a vaccine. And you just go in, you get treated, fixes your DNA, and you're not going to have to worry about those diseases. Still a ways off, but that's where it'll be eventually.
Do you believe that in meetings like MOMED, like this conference here, people are still catching up with where gene editing really is, or they're asking the right questions?
I think that, this is a very sophisticated crowd. I think they understand gene editing, because you really have kind of a couple buckets of therapies here. You've got the cell therapies, you've got the gene therapies, and you have gene editing. And those are kind of the 3 buckets. People are very sophisticated, I think, and understand. And it's interesting, a year ago at this meeting, you had just had Liberation Day, you had, the stock market was really low. It was pretty, overall was not a great time, sort of broadly in the geopolitical realm. I think today people are much more bullish about where we are as a space. There's been a great clinical data. So I think it's interesting to watch the evolution over time, but I think people were very positive and there's a lot of good momentum in the field.
Devyn, thank you very much for joining us and for your insights.
Pleasure.